Hello dear students, welcome to the next session of the principles of inheritance and variation. Today we are going to discuss regarding the next topic, that is the test cross. Before that, so what we discussed in previous class, so by using Punnett square, so we are able to get all the possible probabilities of the gametes formed and then the combination of the gametes which leads to the formation of a different combination of gametes that leads to a different offsprings and their phenotypic ratio and then the genotypic ratio. So from that preceding example, so we are able to get the phenotype of the organism and as well as we are able to calculate the genotype of the organism by using mathematical probability. But see, as the genotypic ratios can be calculated by using the mathematical probability, but just by simply looking at the phenotype of an organism, if it is showing the dominant phenotype, so just by looking at the dominant phenotype of an individual, it is not possible to predict or find whether the genotype is homozygous or the heterozygous. For example, if I give you a tall pea plant, by just looking at its tallness, is it possible to say whether it is a homozygous tall or the heterozygous tall? Because homozygous tall will also show the same height, heterozygous tall plant will also show the same height. How can you say whether it is a homozygous tall or the heterozygous tall? So, just by looking at the dominant phenotype of an individual, it is not possible to find out its genotype. So, say for example, whether a tall plant from the F1 generation or the F2 generation, whether it is having a homozygous or the heterozygous genetic combination, cannot be predicted by looking at its phenotype. So only the Mendel designed another cross that is only called as a test cross. See, this test cross is very important for 2 to 3 marks for the PU examination, and as well as it is very important to answer the various questions, numerical questions for NEET and CET examination. So let's see. See, what do you mean by test cross then? How can you define the test cross? So here, test cross is nothing but, test cross is a cross between, is a genetic cross between a dominant phenotype of F1 or F2 are the individual showing dominant phenotype in F1 or the F2 generation with its recessive parent. So recessive means it will be homozygous recessive only. So cross between the dominant phenotype of the F1 or F2 generation with its recessive parent is only called as test cross. So this one was given by, so it was designed by, this test was designed by Mendel only. So by conducting this test cross, the Mendel was able to find out the genotype of the unknown dominant phenotype of the F1 or F2 individual. Means in test cross, the individual which is showing the dominant phenotype will be crossed with its recessive parent or the homozygous recessive parent in order to find out its genotype, and that is only called as a test cross. In PU examination, you will be asked a question such as define and design a test cross. Then you have to write this definition for the test cross. Then you have to write an example. So make a note of these points. Then we are able to write an example for the test cross. See, this is what an example in order to demonstrate or in order to describe a test cross. You have tried this example. Say for example, here we have taken a flower of the pea plant. So, whose color is violet? So, I have used blue color here, consider it as a violet colored one. So, that colored flower is the dominant phenotype of the pea plant. So, we know colored flower is there, violet color flower is there, means it is a dominant phenotype. So, whether it is from the F1 or the F2 generation. So, it's violet colored means it's showing the dominant phenotype of that particular character. Then what its genotype is, whether it is the heterozygous dominant or the homozygous dominant, we don't know. So its genotype is unknown. That is the question mark. We don't know the genotype of that particular character or the phenotype in pea plant. Then what we have to do? So we need to conduct a test cross to determine whether it is heterozygous dominant or the homozygous dominant. For that, we need to cross that unknown dominant phenotype showing individual with its homozygous recessive parent. So when it is crossed with the homozygous recessive parent, okay, so its homozygous recessive parent will always produce the gametes which will have the recessive allele only, recessive allele only. So there are both possibilities are there. The unknown plant may be having a homozygous dominant condition or heterozygous dominant condition. Let's assume, okay, if it is having a heterozygous condition, a dominant condition means then the gametes produced will be two different types, that is one is with the dominant allele and one is with the recessive allele. Then what happens? What are the progenies produced will be having which type of phenotype? Means the progeny produced will be having here capital dominant allele with recessive, next dominant with recessive, then recessive or recessive, and again recessive. So, if at least one dominant allele is present means it will express its phenotype. So, here the plant is with violet color flower will be produced. Here also the phenotype is violet color. And when homozygous recessive condition is present, it is nothing but here the white colored flower will be present. So, in a cross, see, forget about this part now. When you cross that unknown dominant phenotype with the homozygous recessive parent, if in the offspring generation, if you get the phenotypic ratio, that is half of the flowers or half of the plants are offsprings with violet flowers and half of them with white flower. That is what the phenotypic ratio. So the phenotypic ratio is that is 1:1. 1:1. That is phenotypic ratio is 1:1. And what is genotypic ratio? The genotypic ratio is that is 2:2 or 1:1. That is 2 are heterozygous dominant and 2 are homozygous recessive. See, 2 are with heterozygous condition and 2 are with homozygous condition. So their ratio will be 2:2. Otherwise, 2 ones means 1:1. That will be the genotypic ratio. Then what is the interpretation by this result? Means interpretation is that is the unknown flower or the unknown plant is The unknown plant is heterozygous dominant. Heterozygous dominant. The unknown flower is heterozygous dominant. That is, it is having a genotype in heterozygous condition. See, this is how you can find out the genotype of the individual by conducting a test class. This is one possibility. If you get these results means, then what is the interpretation? Unknown flower will be heterozygous dominant. Similarly, okay, what is the other possibility means? Here the other possibility is nothing but here, if the flower is having, or the particular plant is having homozygous dominant condition, then what happens? Here the gametes produced will be the same, that is both will carry the dominant allele. When they are crossed with the homozygous recessive parent, that is white flowered plant, then what are the possible combinations means here? Dominant with recessive, dominant with recessive, dominant with recessive, and dominant with recessive. Means here all the plants produced are with a dominant phenotype, that is violet flowered. So all are with the violet flower. So all are with violet flower. So what is the phenotypic ratio means here? See, all are with violet flower. All are with violet flower only. Then what is the genotypic ratio? Here the genotypic ratio is, what is that one? One only, or what is that? All of them are the violet. So the all are dominant. All are a dominant phenotype. All are with a dominant phenotype. That is capital and small w. That is dominant allele with the small allele. Then what is the interpretation? If all the individuals of our offsprings produced in the offspring generation showing the same phenotype means, then that unknown individual, unknown plant is homozygous. It is homozygous dominant. It is homozygous dominant. That is, its genotype is capital W and capital W. So if it is homozygous dominant, then only it can produce only one phenotype in the offsprings. If it is heterozygous dominant means two different phenotypes will be produced, or two different traits will be produced for a particular character. So if phenotypic ratio and genotypic ratio is same, that is 1:1 and 1:1, then the genotype of that unknown individual will be heterozygous. If the phenotype is same for all offsprings and the genotype is also same for all offsprings means, then the unknown individual genotype will be homozygous dominant. This is how you have to write or design test cross. So first you have to write the definition of the test cross, then you have to write this example. So it will be asked for 3 marks in the PU examination. Is it clear, guys? Okay, so make a note of these points now. See, from the monohybrid cross, the Mendel, based on his observations during the monohybrid cross, he was able to draw some conclusions, but a perfect or the definite shape for these conclusions was given by another scientist, and his name is Carl Correns. Means from the observations in monohybrid cross, the Mendel simply he drew some conclusions. But for that conclusions, a definite shape, that is in the shape of laws, which was given by the Carl Correns. Mendel simply wrote some conclusions based on the observations of monohybrid grass. But they they were given in the form of or the shape of the laws by Karl Lorenz. So, on the basis of observations in monohybrid grass, Mendel simply proposed two general rules. They are popularly known as or called as principles of inheritance or the laws of inheritance. In that, the first one is the law of dominance. Law of dominance, or simply it is also called as the first law of inheritance. The first law of inheritance. It is called as the first law of inheritance. So what this first law of inheritance says that? See, when Mendel experimented the garden pea plant with 7 characters, each time or in each case he found that only one character, or in each case what happens, every character is controlled by discrete units, and he called that discrete units as factors. Remember, the Mendel observed something which was being passed stably without getting changed from parents to offspring through the gametes, and he called that one as a factors, and now we are calling that factors as genes, right? So that is what he is mentioning here. When Mendel experimented in garden pea plant with 7 characters, in each case what he found means each character or every character is controlled by discrete units, and those discrete units are called as factors. And so what is the nature of these factors? These factors always occurs in pairs only. These factors always occurs in pairs, and that pairs has nothing but alleles, and that pair of factors is called as alleles, right? So the factors always occurs in pairs. And the next is in a dissimilar pair of factors. In that pair of factors, if they are dissimilar to each other, means contrasting alleles are present means. So in a pair of dissimilar factors, say for example the capital T and then the small t, both are coding for the same character, that is plant height, but one code for tallness, one code for the dwarfness. This is called as a pair of dissimilar factors. In such a pair of dissimilar factors, what happens? Only one is able to express its phenotype, and the one which expresses its phenotype is called as a dominant factor, and such factor is called as dominant factor. And the other which failed to express in the presence of the dominant factor is called as a recessive. That is called as a recessive factor. So, as it explains the expression of only one phenotype in the F1 generation and appearance of both phenotypes in the F2 generation. So the one which get expressed in F1 generation is called as dominant, and the one which failed to express in F1 generation but reappears in F2 generation is called as a recessive factor. So the law of dominance explains the monohybrid cross, and it also explains the ratio of monohybrid cross, that is phenotypic ratio of monohybrid cross is 3:1. That is 3 are found to be with dominant phenotype and 1 is found to be with the recessive phenotype in F2 generation. But this law of dominance is not universally accepted. Remember, this is very important one for the NEET examination. So this law of dominance is not universally accepted. This one is not universally accepted. So why it is not universally accepted means always there will not be a appearance of the dominant phenotype. There are some examples are there in a genetic class where none of the parental phenotype will be expressed, but as a mixture of them the intermediate phenotype will get expressed. That we are going to discuss in the next session. So only the law of dominance is not universally accepted. Which of the law of inheritance is not universally accepted means that is law of dominance. Make a note of these points now. Later we will discuss regarding the second law of the inheritance. So the second one is the law of segregation. So what it explains means, see, this law is based on the fact that when two factors are present in a single individual for a particular character, so when two dissimilar factors are present for a character in an individual, so those dissimilar factors never get mixed up or blending, and both of those traits or the factors get recovered as such in F2 generation, even though one of the trait which is not seen in the F1 stage. Say for example, the homozygous tall plant, and then the homozygous tall plant, and then the homozygous dwarf plants were cross beans. The particular trait, even though the zygote contains dissimilar pair of traits, that is one coding for the dominant allele, one coding for the recessive allele, but the plant will develop as tall only, but dwarfness will not be seen in the F1 stage. But when such plants are allowed for the selfing, in F2 generation, both tall and dwarf plants will be produced, right? So, even dissimilar pair of factors are present for a character in an individual, even though one of the trait is not seen in the F1 generation, but both will be recovered in F2 generation as it is as such. So, why it happens means, so why one of the gamete will not be, or one of the factor will not get expressed in the F1 generation, but both of the traits will get expressed in the F2 generation means, the reason is, during gamete formation from the parental plants or the parental individuals, the factors of a particular pair separate and segregate in such a way that a gamete or each gamete receives, or one gamete receives only one factor for all or for each character. Say for example, 23 characters are present, traits or the factors will be present in parental generation. For each character, one factor will be received by that gamete. Means it receives 23 factors only. Like that. So that this ensures the purity of gametes. This ensures the purity of gametes. Let's understand with example. Say for example, the plant is homozygous. If you take the example as plant height, if the plant is homozygous, means it produces similar type of gametes, means only one type of gametes which are carrying the same type of factor. If the plant is heterozygous, then what happens? It produces the gametes which are having the alleles which are dissimilar, or two different type of gametes will be produced. 50% of the gametes will carry the dominant allele and 50% of the gametes will carry the recessive allele in case of heterozygous individuals. But if the individual is a homozygous, means all the gametes produced will be of same type or similar to each other. So only, as this one is universally holds good, so law of segregation is universally accepted. So this law is universally accepted as it is applicable for all organisms present on the earth, and as it ensures the purity of gametes, the law of segregation is also called as the law of purity of gametes. Is it clear, guys? So based on the monohybrid cross observations, the Mendel proposed two laws of inheritance. One is the law of dominance, and second one is the law of segregation. Make a note of these points now.